Hi, thank you so much for stopping by to check out my poster. My name is Anjali Verma and I'm a senior in STEES studying applied physics. This summer I got the opportunity to participate in the Columbia Department of Biological Sciences SURF program. I worked in the lab of Dr. Ruben Gonzalez studying the mechanisms by which ribosome conformational dynamics regulate peptide synthesis. As I'm sure you will all remember from Moush Bio, or maybe even from high school, ribosomes make proteins. Yet, while ribosomal translocation is one of the most fundamental processes in biology, the conformational changes that underlie this process remain poorly defined. Mapping such changes in ribosome structure would advance the development of next-generation small molecule therapeutics that could neutralize dysregulation of protein synthesis, which is a hallmark of cancer and many different viral infections. It's clear that greater insight into ribosome conformational dynamics is critical for the design of new inhibitory drugs that specifically target non-canonical translation events caused by disease. So now that we know kind of the big picture, let's zoom into my specific research. The Gonzalez group where I worked over the summer is a biophysics lab that conducts a lot of single molecule experiments which allow for really fine resolution and detection of molecular motion. One such technique is single molecule FRET, or Forster resonance energy transfer, which I'll refer to from now on as SMFRET. So SMFRET works by using two fluorescent dyes whose emission and absorption spectra overlap. This way, when the dyes are close enough together, you get an energy transfer and you see a little burst of light. This summer I was able to use Python, because we were all at home during the coronavirus lockdown, to design these sorts of experiments, which really meant I was able to pick the best places on the ribosome to actually put these dyes. To do this, I comparatively analyzed two different cryo-EM structures of yeast ribosomes. One was in the canonical state and one was in the hybridized or rotated state. And with a little linear algebra and some SciPy, I was able to determine what donor and acceptor positions on the ribosome would be best for future experiments. I wanted dye placements that would result in really bright FRET signals, but that were also able to describe the important conformational changes of the ribosome. So the relationship between the brightness of the light emitted by the FRET pair as the distance between the dyes is pretty well characterized through a concept known as FRET efficiency. So I was able to use this formula on existing cryo-EM structures to predict the brightness of future SMFRET signals. I wrote a Python script that was able to do most of this, but I still had to take the time to actually visualize my FRET pairs using PyMol. So let's wrap up. This summer, I was able to use Python to analyze cryo-EM structures of eukaryotic ribosomes in order to select the most interesting FRET pairs for future experimentation. The FRET signals I identified this summer maximized FRET efficiency while still reporting on functionally significant conformational changes of the ribosome during the elongation phase of translation.